Okay, we continue with our course, Pharmacology and Physiology of Neuropsychiatric Disorders. And as we discussed, it's our 5th meeting, and it will be interactive visit to the Neuropharmacological Laboratory Center of Biosciences, Slovak Academy of Sciences. There is a small change, and I see it's good change. It will be visit not only to Neuropharmacological Laboratory, but to the whole department of biophysics and electrophysiology, to my colleagues' laboratories and my laboratory. I am broadcasting from this beautiful Patronka campus of the Slovak Academy of Sciences. Slovak Academy of Science, it's unlike Western Academy of Science like French or British, which are only learned societies, and or Israeli Academy of Sciences, which are learned society and body which provide research grants. Slovak Academy of Science, it's a huge network of the actual governmental public research institutions and centers, something like similar to the American NIH, National Institute of Health. But Slovak Academy of Science institutes, they not only about medical science, it's about all areas of science. We are divided to 3 departments. First department is mathematical, physical, and computer sciences. Second department is biological and chemical sciences. And third department, social and humanitarian sciences. And each department has multiple research centers and institutes. And we are now located in the Center of Biosciences of the Slovak Academy of Sciences. It's one center, but it's located in 2 cities on the opposite sides of Slovak Republic. In Bratislava, national capital, we have 2 institutes belonging to the center: Institute of Molecular Physiology and Genetics, and Institute of Biochemistry and Genetics. And on the very beautiful city Košice, in the eastern Slovakia, we have another institute. It's the Institute of Animal Physiology. And this is how what type of research is performed in our center in our Institute of Animal Physiology and Genetics. We have Department of Biophysics and Electrophysiology, which you will visit today. Our department dealing with neuro and cardiophysiology. In addition, we have Department of Biochemistry and Cytochemistry, which is primarily focused in the cancer, or more specifically, leukemia research. Another institute located in Bratislava, Institute of Animal Biochemistry and Genetics, they are dealing with also with the neurophysiology and ethology, but where our institute primarily used rat models, they working with the avian models, with the birds. And they also have pretty interesting department of biochemistry of biological membranes. And finally, our institute in Košice, they have 2 departments. One is one focusing in the gastroenteric physiology, and second is very interesting department focusing in reproductive and developmental physiology. This, as we have plenty different area, multidisciplinary research, open very good background for collaboration. For example, we are collaborating with Department of Developmental Physiology, and we investigate together how maternal stress and maternal depression can affect development of the brain, of the embryonal offspring brain. Today we will be mostly in our Institute of Molecular Physiology and Genetics. In Department of Electrophysiology, we have 6 labs in the department: biosensors, cell cultures, electrophysiology, ion channels, neuropharmacology, which is actually my lab, and confocal stat microscopy lab, And today we will have, we will see, you will meet my dear colleagues, you will meet Dr. Katarina Ondacheva, who will show you cell cultures, patch clamp electrophysiology, and STED microscopy. Doctors Marta and Yana Gabryakov, who will show you a laboratory of ion channels. Danil Grici, my postdoc, and my PhD students, Ruslan Palioha, Tala Khoury, and Hande Oshbasak will show you and myself, we will show you my laboratory of neuropharmacology. Okay, I'm stopping for a while. Stop sharing, and the floor will be now off with Tala and Hande, who will be our camera people today. They will walk around. They will show you the laboratories, and I will continue to comment from my place here. I am stopping screen sharing to allow them to work with the camera more efficiently. How to stop screen sharing? Did I stop it? No.
No, you're still screen sharing.
I see everything. I see how to do it. Yes. Okay, I stop screen sharing. Tala, please.
Tala, please.
Good morning, everyone. My name is Tala, and I will be guiding you through our wonderful facilities today. I am a PhD student working with Dr. Drumenko on the neuropharmacology lab. And now I will take you around to show you my colleagues and their exciting research that they will be leading. Come with me.
Sorry?
Yeah, the light.
Okay, wait,
Okay, let's go.
We have sometimes when they walk on the floors, we will have freezing picture, but don't worry about it.
We are here. Yes, we are changed to Wi-Fi networks because maybe partial freezing. I'm sorry for technical.
Yeah.
Yes.
You can speak to our
Yes,
students.
Dr.
I would like to say nice to see you, but we can't see you. But I hope you will enjoy this live seminar with us.
Katka, introduce yourself first. This is Dr. Katarina Ondacheva.
So hi, so my name is Katarina Ondacheva, and I work in this department as a molecular biologist and electrophysiologist, and I mostly focus on hippocampus. I prepare primary hippocampal cultures as well as brain slices. So it's my introduction.
Okay.
Hello, my name is Hande and I'm PhD student and I'm working with Dr. Elia Hudramenko. And now that's all.
You will show us around?
Yes,
so-
let's go to see
Yes.
this lab, electrophysiology lab, right? Come with us. So here is our electrophysiology laboratory. You can see. So we are very proud to have it. We have these two patch clamp units, and for sure you already know this patch clamp.
We have freezing
Yes,
a little bit.
So,
Yeah, now it's okay.
it due to
Yes,
it should be this network maybe.
it's frozen in second floor. Just a moment. I hope they will be back soon.
We are here. We are here.
Can you hear us?
Can you
Yes,
hear
I
us?
hear you, but the picture is still frozen.
Okay, so it will be maybe difficult. Here is like a bit weird network.
Here. Now better.
Maybe.
Not really.
Maybe
Okay.
go to the back.
Maybe come back to it.
Okay. So we have this special apparatus. This one is used for primary cultures, and the other one is used for brain slices. So we are able to cut the brain in thick slices, and we can measure single cell inside its natural environment. So as you can see, a patch clamp apparatus consists of microscope, inside Faraday cage, anti-vibrational table, and we have already prepared cells. So I can maybe try to patch one if it will work. Let's see. So first of all, I forgot to mention that we have to prepare pipettes, which are made from borosilicate glass. It's made over here with this machine called cooler. So we have to switch it on. We install program. And here are borosilicate glass pipette. They look like this. They are very thin. They are very thin. I'm not sure if you can see it. So this puller uses 2 temperature cycles in which he split this one pipette into 2 pieces. So it will be really, very thin in the edge. You can see now will be the first step. And in the second step, it will be split. And the second one right now. So you can regulate, it depends on what type of cells you are going to patch. You can regulate the size of the pipette tip. It's not sharp, because when it would be sharp, now it's, you can see. It's like this. No, you can maybe see it's really very thin. So use such a pipette, such pipettes for measurements. Okay, so let's go to a microscope. The first, I will tell you principle, but maybe you already learned it. So the first of all, we find a cell. We touch the cell pipette. Here inside the pipette is intracellular solution and electrode, which is recording every electrophysiological parameters. When we touch the cell membrane with a small suction, we just make really tight junction. It's really tight between pipette and the cell membrane. And with a really strong suction, we can disrupt the cell membrane and we can record. As the plasma membrane has different type of channels, calcium, potassium, sodium, so you can measure different types of currents produced by the cells. So it's like this, yeah. Go.
Is there an issue with the network? Or can you hear us and see us?
Are you see us? I'm sorry, you see us or not? Boys and girls.
Yes, it's
We
okay.
see
We
you.
do.
I'm sorry, please say.
It's frozen, yeah.
Because it's
Yes,
frozen.
yes, it's okay. I will tell you if some problem.
Okay.
So now we have this pipette which we prepared, intracellular solution which we put inside the pipette. It's not visible very much, but so, and unfortunately, yesterday in the evening, our computer, where his camera broke, so you are not able to see what I see inside microscope. But I just put picture, very simple picture here, how it looks like inside the microscope. So I have this pipette, I find my cell, and I try to go above the cell. So I'm going to do it right now. Yes.
You have to try me to see.
I try to see the cell.
Yes.
Yes.
You try
to see with pipette, right?
Yes. I try make it full. And after I try to show from where.
Okay, let's see.
Camera, directly from the camera.
If we can see it. So now I have cardiomyocytes. I have not prepared hippocampal cultures. We had not PAPs prepared. But now you can see. You will see cardiomyocytes with the pipette. And the pipette will be very close to the cell. So you can try to see.
I'm sorry.
You can see it. You can try, or when it won't be visible, I'm not sure it will be visible.
Yeah, right.
I'm a bit afraid that you can't see it well.
No, not really.
I'm sorry.
No.
Just a moment. No. We will see.
This popular, I can put it somewhere. Oh, it's okay.
No, Daniel, you can stop it. We will. Yeah, maybe. Yeah, we see something now. This is cardiomyocytes.
Yes, and you can see the
And this is a
pipette.
pipette. Yes, we see it
Yes,
now.
exactly. And now I'm going to touch the cell and I will try to open it if it will work. So we will see it in this monitor.
Yeah, now you can see it.
Now you can see it well. Okay, perfect.
I'm sorry. I cannot make more. I'm sorry.
Okay.
It's okay.
So you can see it, how it looks like. And now I'm going to touch the cell. I'm trying to touch the cell, and you can see it then on this computer. We will see it here on the monitor.
This is a potential recorded. When
Yes.
Katka will touch cells, you will start to see the change in this potential
Yes,
immediately.
exactly. You will see the line. This line will go up. So it means that I touch the cell. So let's see. I'm going. Wait, and I will try. And now it's going up. You can see, right? Now I touch the cell, and now you see there is tight junction between the cell, and now it opens. So this peaks mean that the cell is already open. So
Electrode penetrated
really
inside of the cell.
Yeah, it was really fast. Even I was not prepared, and it opened. So let's try to see potassium currents. If we will have some recordings of potassium currents. So we are changing membrane potential of the cell. We are stimulating the cell and changing her membrane potential. You can see it here. And here is recorded current. It has, I think, 13 or 15 steps, this protocol. And we will see if there will be visible something or not. Now it's not so nicely seen. Nothing, maybe the cell was not so okay. So the currents, I can't see well. I can't see, but I can show you recording I have. Finish or not? So it seems like this. Yes, the curve should be like a little bit more up and a little bit more down here. So the recording will stop. And so it's done. So this is our recording, but I can show you how it should look like when you are lucky. So maybe I will try this cell. Let's try. It's a little bit more like up and it's going a bit more down. So the recording looks something like this. Wait. And here it will be even better visible. Yes, here it's better visible. So this potassium current. And as the cells are excitable, neurons are excitable, we are focused also on action potentials and on action potential series, as this excitability is really, really very important for a cell to communicate and to like have a proper functions. So here we are doing with the slices, so you can see camera with the cells. And I can show you recording of hippocampal neurons in slices. So like this is going to generate single action potential. Now it's going to this peak of action potential. You can see it. So this is first recording, and action potential series looks like this. You have several peaks. So the cell is really excitable.
This is a burst that we discussed in our classes several times.
So and with this patch clamp technique, you can really see different type of current channels, and you can even with this perfusion station, you can study pharmacology. So you can apply different compounds during the measurement. So you can see acute effect of this compound, of this your pharmacological compound. And also you can exclude different currents. For example, you don't want to measure calcium currents, you want to focus on potassium. So you use different types of blockers, toxins, conotoxin, tetrodotoxin, or different types. So you are specialized only on one type of currents. So, and that's it, that's patch clamp, which we have here. Maybe I can show you even how it looks like, one more picture, how it looks like hippocampus, the hippocampal cells, you can see them in a layer of a tissue. So we are patching the single cells. The single cell is just picture how it looks like in a tissue of hippocampus. That's it. And here you can see nicely whole hippocampus, which we have pictures in here. So it's like this. So now I introduced you a little bit of this technique, and we can move forward to the confocal. So we are also working with confocal microscope. So it's next to the door. So it will be fast in here. So we have this Leica microscope, which is like this.
We are a little bit
It is so
frozen.
amazing, and we are proud to have it. You can
Frozen.
No,
Just a
yes,
moment,
it is-
please. I'm sorry, everybody talks. Just a moment. I'm sorry, Yanka. You have password from Yanka network or not?
You can open the door, maybe you can just.
Yes. Eli.
Yes, it's bad. No, but we don't hear now.
You hear us or not?
Yes, I do, and I see.
You hear us or not? Katka,
Yes,
listen.
Yes, can you hear me?
yes,
Okay,
yes.
Because I don't know how it's gonna network, so this is the high resolution microscopy, so we are able to see different structures of the cell. You can see gene expression of the proteins of interest. You can see visualize different organelles and the plasma membrane channels, whatever you really want. So we are proud of this computer, of this microscope we have here. So first of all, when you want to see something, you have to prepare your sample. So if it's like this, you have your glass, microscopic glass, and here inside is cover slip with your cells, which are fixed. Cells has to be first fixed, and then you need to like permeabilize the membrane if you want to visualize something inside the cell, or you don't have to permeabilize the membrane. Then you use primary, secondary antibodies, which secondary antibody is photogenic, so it can upon excitation, it can emit fluorescence. So we put this here, in this holder.
Can I see?
It depends on what you want to see. We have objectives which are for water and which are for oil. It depends on what you want to see. So we close it like this. And when you switch laser, you can see the laser from maybe like this. And from side, you can see the laser blue one.
Right.
There is laser which is accepted by our sample, and it can emit fluorescence. So and now we can see different structures. Mostly I'm focused on neurons, so I stain them with MAP2. So I can visualize whole hippocampal neurons. And this is really, really good when you want to see neuronal arborization, what means branching of neurons, when you want to see how are formed dendrites and axons. So then we are also seeing different channels and different type of proteins inside. For example, here our colleague is working with cardiomyocytes. Cardiomyocyte, you can see it here, cardiomyocyte, and she stained it with, she visualized reacting with oxygen species. So then we can see visualized nucleus inside of cardiomyocyte. And you know already about cell division. So we could visualize using tubulin, like division of the cell. This is prophase NFAs.
where you can see that the cell is going to split into pieces. Here I visualize nucleuses. And it's the same, yes, cell division. Here it's already divided. And you can do also colocalization, which means that you can, in the one same sample, one cell, you can visualize different type of proteins that are colocalized together. Here we can also see, but it's not fluorescently stained, egg with the sperms. So it's embryo, going to be embryo. So this microscope is really used in our department to study different structures. And that's it, I think. I hope you understood and you enjoyed. So, see you. Bye.
Katka, maybe you can show briefly in the corridor. There are very nice methodological posters. It will be
Yes,
sharpened.
okay.
Yes, posters about
Your
Kung Fu. No, this is, yes.
Which one?
Yes, you can
Show
see like
Page
this
club.
everything. We have
Page Club
Club,
and
Great.
yes, there is very nice Page Club poster.
This is confocal.
Here's which one.
Just a moment, Page Club. Just a moment, please.
Yeah, it show different Page Club techniques here.
Okay, yes, of course. You see it?
recordings, maybe you can see.
You see it? Different Page Club techniques.
Yeah, this is exactly how what yes, full cell. Yes, this
Yeah.
is
And this is different record.
Current.
Different.
this is
different,
current records. In Page Club electrophysiology, we have two possible modes. One it's called current clamp. When you hold the current, when you set up the current, and you record the voltage response of the cell. And second one is a voltage clamp. It's other way around. When you hold the membranal potential and record different currents. Actually, today Katka showed you both of them.
Okay.
Okay, next I think Hande will show her recent results from
Yes, 400.
Right.
Thanks. Thanks.
Hello.
Hello,
Hello.
Hande. Did you put your poster about very
Yes.
good.
Yeah, this is my poster. So I will talk about my PhD topic, and I will give you quick information about my PhD topic. So I'm basically working with agmatin. And what is agmatin? Agmatin is natural compound. It can be found in our body, and it is made from amino acid and L-arginine. And why we interested with agmatin? Because agmatin has shown very excited effect on mood regulation, especially for brain. When we look at the papers, we found so many exciting paper about it. And then in my topic, we want to see and how agmatin is works in dorsal nucleus, especially for a rat. And if I say, for example, previous research has indicated the serotonergic neurons shows shows an anxiety and antidepressant-like effect. And in our method, we did two main experiments. First one acute, second one chronic treatment. For acute treatment, we want to see initially immediately effect with single dose of agmatin treatment. That's why with electrophysiology method, we recorded 5-HT neuronal firing activity. With we administrated a cumulative dosage of agmatin 1 to 3 milligram per kilogram, and we found that there is increased dose-dependent increase in the 5-HT neuronal firing activity, and we also applied chronic treatment 14 days. And we found that again there is a significantly increase in the agmatin treated rats. And we also wanted to see this agmatin affects via which receptors. That's why we applied two different drugs. First one is mostly known escitalopram. So escitalopram is SSRI. So it means serotonin transport inhibitor. And another one is WAY. This is 5-HT1A receptor antagonist. So we, after chronic treatment, 14 days chronic treatment, we applied escitalopram. And then we saw there's a decrease, both agmatin and both control group, there's an increase. But when we treated with agmatin, increase a little bit higher than control group. When we used 5-HT1A antagonist, we couldn't see, we saw very small recovery, but we couldn't see recovery like control group, as you can see here. So what is our conclusion and what is our result according to these experiments? So agmatin has agmatin and anxiolytic and antidepressant effect, but in this experiment we saw that agmatin's effects are likely mediated through the stimulation of 5-HT neuron in dorsal root nucleus. And our second conclusion is agmatin also, I forgot to mention about that, agmatin can inhibit nitric oxide synthase. That's why our second conclusion is agmatin actions as a nitric oxide synthase inhibitor may play a crucial role in serotonergic transmission. And for our last conclusion, between neuronal nose inhibition and 5-HT1A autoreceptors, there is a potential interaction between them, and agmatin's unique effect can be related with this 5-HT1A receptor and nose inhibition. So our results open new avenues for understanding complex interplay between nitric oxide and serotonergic system in mood regulation, and potentially can lead to novel therapeutic strategies for anxiety and antidepressant-like effects. And thanks so much. I'm so excited.
Okay.
Thank you,
Alright.
Tala. Thank you, Handa, sorry.
Thank you so much, Handa. So exciting. Now, let us show you something in action and live, and I'm going to take you to the animal facilities. So come with me. All right.
Okay, now you see me here. I will show you a little bit posters from our research. Now let's start from something.
The camera?
let's-
The camera is the other
it's,
way.
I'm sorry, yes?
The camera is vertical instead of horizontal.
My camera?
yes, you start to be vertical.
Just a moment. Okay. Now it's better.
Okay, yes. Thank you.
I'm sorry. Okay.
It's our recent project. It's we will have class about it, a license class. Our class will be dedicated about interaction between growth factors and monoamine system. Here we saw effect of FGF2 fibroblast growth factor on dopamine neurons, and we found that FGF2 stimulate dopamine neurons, and antagonist of FGF2 receptor 1, its G protein counterage, has an opposite effect, and this effect has and it was another experiment we also checked for serotonergic neurons, and serotonergic neurons we found other opposite effect. It's decrease the activity of serotonergic neurons, their burst activity. And this is my project actually. Here you can see your university, Tel Aviv University. This is my project with Segev Barak, and we will discuss it in one of the last classes. It's our another work. We worked with effect, or we also have class dedicated to this. It's about interaction between delta opioid receptor and hippocampal and serotonergic neurons. We will speak about it. And now I would like to introduce to you, we have a scientific journal in our institute. It's pretty good journal. It's a journal indexed in Scopus Web of Science, and you are very welcome to, you and your supervisors, welcome to submit to us, and I will meet you with our technical editor. Hello, Vera. Dr. Vera Kominkova, my researcher in my lab, and she is a technical editor of the journal.
Welcome to our editorial office. My name is Vera Kominkova. I'm technical editor, and my colleague Eva Krejčeva is on holiday. Yes. We have a website. Our journal is.
We can't hear her very well.
It's okay.
I can hear
sorry.
the guy holding the camera, but I can't hear her very well. If she can just speak up a little bit. Thank you.
Yes. Our journal, General Physiology and Biophysics, is an open access, peer-reviewed scientific journal. It is published by our institute since 1982, and we publish original research articles and reviews. We publish results from physiological, biophysical, and biochemical experiments at the cellular and molecular level. We have also electronic publishing system, MM+. You can see on this website. Yes, you can see articles and all dates of journal. Yes, this is date, published, author, decision, and this one. Yes. On our website, you can see instructions for authors. Yes, these submissions.
This is a paper
Would you
copy.
please rotate the camera?
I'm sorry, once again.
Could you please rotate the camera? It's vertical instead of horizontal.
I'm sorry. Oh my God, once again. Okay.
Thank you.
I'm sorry. It's okay.
Anyway, if somebody interested to publish, ask, contact me. I'm a deputy editor. I am deputy editor in chief. This is our paper copy. Our impact factor, unfortunately, still need to grow up. It's 1.5, but it's still the recent impact factor, recognized impact factor, and this journal is open access, and we offer very fast peer review. If somebody really need to publish fast data, we always do use service, and this is a paper copy of the journal. So if somebody interested to publish with us, please ask me. And now you go to visit another lab. Before coming back to in vivo electrophysiology again, you will go to visit to the most amazing lab I will like seat of Dr. Jan and Marta Gobriakova. I go back to my office, and Daniel and Tala will take you to Dr. Gabryakov's.
All right, now let's go to another exciting place to show you another experiment in live action, or to lead you more about the lab that they are working with. With me? In this room, next part. Hello. Do you mind introducing yourself, please?
Yes, Marta, come please. Hello, my name is Marta Gabriakova. This is my colleague and my sister, Jana Gabriakova, and I am the head of the lab of intracellular ion channels. And we use one very special method. It's the electrophysiological method, which we call planar lipid membrane method, or we use the abbreviation BLM because it's like lipid membrane. And now I will show you how we use that. Here is the BLM setup.
We lost the video signal. Now it's okay. Yes.
What we use in our work. It's very simple. Here is the Faraday cage and the anti-vibration table. The anti-vibration table, because we measure the very small currents, picoampere. It means that the electrical and mechanical noises from outside should be eliminated. Then we have the measuring chamber, which is very important. And here is the cup.
We lost video signal. No, okay, you can continue. Yes.
From polystyrene, and in the wall of this cup there is the small hole, which is important for forming the BLM, the black lipid membrane. And I insert this cup into the chamber. Then I fill this cis and trans solution with the trans and cis and trans department with the solutions, and then I will make very easily the membrane by using the lipid solution and by painting this lipid solution over the hole in this cup. It's very easy. It seems easy, but it's not so easy. It's very difficult. The experimentalist has to be very patient, and also manual skill is very, very important. It means that this method is not for everybody. Okay, now I will show you.
Oh my God, I'm sorry.
Yeah, it's okay now. You get frozen. Sorry.
When I found the lipid solution, there is a lot of lipid molecules here, and spontaneously the black lipid membranes self-organize. It means that this is the model of the biological membrane. This process is spontaneous. And then we apply to one side of the membrane the isolated vesicles with ion channels. And then we see we need to induce the fusion of these small vesicles into the BLM here in this hole of this wall. And after the successful fusion of these vesicles, we can measure the current through these ion channels. And this is the animation of real current recordings. We studied the one intracellular ion channel, which is important for contraction of cardiac and skeletal muscle cells. We call it ryanodine receptor. Maybe you know it. It's a very big ion channel, and usually it conducts calcium in the cell. And here you can see the current which flow through the channel. Here the channel is in closed state, and suddenly it opened and it jumped to the open state. Here the activity of the channel is very small, or very low. But when we add some activators such as caffeine, it's very good activator, we can see the activation. It means that the channel jumps more often to the open state, and it's less in the closed state. When we increase the concentration of caffeine, then the channel dwells for a very long time in the open state and goes to the closed state very rarely. Caffeine is very good activator, but is not inside the body or in the cell. The physiological activator of the ryanodine receptor is the calcium, intracellular calcium. When we add calcium, the channel again activates.
We lost video.
Once again, I'm sorry.
Yes, it's okay. Yes.
Is that when we add calcium, the channel again activates. It means that goes more often to the open state than in the closed state. When we add more calcium, the channel again, the channel again dwells for very long time in the open state and goes to the closed state for a very short time.
Right.
And when we incorporate ion channels into planar lipid membrane, we don't know whether the channel is ryanodine receptor or, for example, a chloride channel or potassium channels. Therefore, it's important to have some test that, okay, the channel which I see in the membrane is ryanodine receptor. And for this, we use ryanodine. It's the plant alkaloid. And when we apply this ryanodine to the very active channel, then we suddenly see this effect. It means that ryanodine locks the channel into the subconductance states. And this is the specific test that, okay, I have in membrane ryanodine receptor. Okay, and then I will show you.
Sorry.
I forgot to show you better our method. I'm sorry, it doesn't work. Okay, it means that before we incorporate the ion channel into planar lipid membrane, we need to isolate it from the cell, because this method is used for studying the function of intracellular ion channels. It means that first we need to isolate it from the cell. From this, we use the classical ultracentrifugation, and we isolate the ryanodine receptor from heart, but it also is in the brain. It means that we also successfully isolated it from the brain. And this final pellet, what we get, is here is the electron microscopy picture of this isolation. It means that is the suspension of spherical particles, which contains in the membrane the proteins. Here we can see these black spots are proteins, and some of them are ion channels, because not all of membrane proteins are ion channels. And these vesicles, I told you before, we added to one side of the planar lipid membrane, and then we induced fusion of these vesicles with this planar lipid membrane. It's also very actually, this fusion is coincidence. Despite we use some strategies to induce this fusion, they are not every time successful. It means that we have to be very patient if we want to get a fusion of these vesicles with planar lipid membrane. And when this fusion is successful, then we can measure the current flowing through the channel. And this is the main principle of the method of planar lipid membrane. Now we study, actually, this method can be used to study all intracellular ion channels, calcium channels, chloride channels, potassium channels, but you need to find the appropriate isolation of membrane fractions which contains this or your protein or ion channel of your interest. But now we focus on studying the ryanodine receptor, which is important for contraction of cardiac and skeletal muscle cells. Without this channel, the muscle cells will not contract. And this channel is also expressed in the brain because calcium signaling is also in the brain. However, the specific role of this channel in the brain is not understood as in the skeletal muscle and cardiac muscle. And it has been shown that the ryanodine receptor is the main reason or the main culprit for example, stress-induced arrhythmias. I don't know if you are familiar with this topic or with this disease. There are arrhythmias which are induced during emotional or physical stress. Only during stress conditions, it means that in the rest the patient is okay, but during the stress, the patients can experience arrhythmias, which can be lethal. Okay, it means that the patient can die. And the main reason for these arrhythmias are the point mutations on the cardiac ryanodine receptor. Till now, we know more than 200 point mutations on ryanodine receptor. You can look here, there. And but each patient has only 1 point mutation. Here, done. Only 1 point mutation. And these mutations, when the mutated channels were studied using the BLM method, it has been shown that these mutations cause or make the channel more sensitive to activators such as intracellular calcium, which entry the cell through the other calcium channel, which is voltage-gated calcium channel, which is located on the plasma membrane. And it means that the channel is also active during the diastole when the channel should be closed. And this activity causes the cardiac arrhythmias during the stress conditions when also this channel will be phosphorylated, and this phosphorylation and plus this mutation can cause the dysfunction of the channel. Okay, that's it.
Thank you very much. I would say that it's to my opinion, it's one of the most exciting, sophisticated techniques, because you really record in real time individual ion channel, and you see this quantum states open and closed. For me, it's something really fantastic. Thank you, Mark.
we can measure the function of one ion channel, one ion one molecule. Yes.
Okay.
Okay.
Thanks.
Thank you very
Bye.
much. Thank you. And now we are going to go back to the animal
Sorry,
facilities.
say that we have some technical some
Yeah,
pause for.
Okay, then thank you to I will put screen sharing for a while back. No. Okay, so I would like to thank everybody who helped us today with this guided tours. To Katarina, Katka, Marta, Yana, Vera, Danil, Ruslan, Tala, and Hande. And thank you very much. And we meet again next week on Tuesday at 9:15 in Tel Aviv. It will be regular class. It will be regular class in person, and we will speak next time against our new updated schedule about trace amines. And I will send to everybody soon instruction about final projects. I already get final projects. I strongly repeat, it's not mandatory, but I strongly recommend to everybody not only to submit written project, but also short presentation, which can be done in person during one of the next classes. Or it can be also recorded and sent me and will be uploaded to the course, to our course online. Do you have any questions today?
No, but it was very interesting. Thank you very much.
Thank you, and thank you to more thanks to my colleagues today, because they did excellent work. And okay, thank you very much. See you in Tel Aviv next week. Bye to everybody.
Very much.